14, Mendelian Genetics, is kind of about how we have the information we do. It's called Mendelian Genetics because it was kind of pioneered by a guy named Gregor Mendel, who was a monk, worked in the garden of a monastery, and he was really curious about why things look the way they do. He was super focused on heredity and breeding. And Mendel bred garden peas. The reasons he did this was because, for one reason, garden peas have very distinct characteristics. The flowers are either purple or white or something easily observed, and they're easy to control their mating. And lastly, they're easy to self-pollinate, meaning that one flower can actually reproduce by giving its own pollen to its own female parts and produce offspring in that manner. And in Mendel's experiments, he wanted to start doing some breeding, but what he wanted to do was start with parents that we would call true breeding. So he wanted to start with parent plants that, if they're true breeding, what that means is that they self-pollinate, where they take their male pollen and introduce it to their female eggs. It will produce offspring that are completely identical to themselves. That way, he knows what the offspring should look like before he actually crosses anything. And in Mendel's experiments, he had his P generation. Well, we have these generation names. The P generation typically is described as the parental generation of true breeding parents. So what Mendel did here is he crossed two true breeding flowers. This purple flower, if it were allowed to self-pollinate, would produce more purple flowers, and this white flower, if it were allowed to self-pollinate, would produce more white flowers as well. So he took the P generation of these ones and bred them. The second generation is what we call our F1, or our hybrids, where we have a combination or the offspring of these two true breeding plants. And in Mendel's experiments, when he bred these true breeding purple with white, typically most of the F1 hybrids were purple as well. The F2 generation is what we get when this offspring, the F1, self-pollinates. So this plant right here is going to self-pollinate and produce offspring from its pollen to its eggs. No mixing otherwise. You have two original parents, they're bred together, that offspring is going to self-pollinate and produce offspring, and that is what your F2 is. And Mendel actually saw a lot of different traits in these pea plants. He saw flowers could either be purple or white, they could have flower position at the top or the side, they could have seeds that were either yellow or green, seed shape could be round or wrinkled, the pod shape could be inflated or constricted, the pod color could be green or yellow, and the stems of the pea plants could be tall or short. So he knew a lot of what the traits were and what they should look like when he bred them as he went through. Now, if you think back to our activity where we talked about homologous chromosomes with our whiteboards, we said that we get one set of DNA from dad, let's call it red, and one set of DNA from mom, let's call it blue. Well, when we have our homologous chromosomes line up, they have something called crossing over, which occurs sometimes where DNA will be exchanged, little from moms to dads and vice versa. Well, these homologous chromosomes, we said, were chromosomes that code for the same trait or the same physical feature. In this case, we're talking about flower color. So we have chromosome here, this blue one, call it chromosome 1 and chromosome 2. These are homologous in that they both code for flower color in our pea plants. And what we see here is that along these two chromosomes, 1 and 2, there exists an allele, which is a different version of the gene. So flower number 1 has chromosome number 1, which gives it purple flowers. Okay? The locus is that location of that gene that codes for purple flowers. Well, along chromosome number 2, the locus is in the same spot, but it has an allele, and that this section of DNA actually codes for white flowers. So what you see is that along homologous chromosomes, the locus is located in the same spot, but it might be a different version, whether it's either purple with a capital P or white with a lowercase p. The locus just signifies where along the chromosome that gene is at. And in Mendel's studies with pea plants breeding here and there, he actually came up with 4 fundamental concepts which are kind of the basis of all genetics. The first one is that in breeding, variations exist among different alternative versions of genes. It could be purple, capital P's, or it could be white flowers with lowercase p's, but there's variations in characteristics because there's different versions of the same gene. The second idea is that for each character, an organism inherits 2 alleles, one from each parent. So parent A has an allele it gives you, and parent B has an allele it gives you, but you inherit half of your DNA from mom, half from dad. The third idea was that typically, if the 2 alleles differ, the dominant one is typically going to determine the appearance. So in the case of our flowers, if you get a capital P from one parent and a lowercase p from the other, the capital P is dominant and is going to express over the lowercase p, and we'll end up with a purple flower. Typically, dominant is more powerful than recessive in terms of expression of traits. And the fourth idea we talked about a little in meiosis is that when you have meiosis occur, you have 4 daughter cells that are different from the parent, and they can inherit the DNA from mom and dad in any sort of fashion. So this one could get a blue, could be a red over here, could be a red here. It really could be wherever. It doesn't really matter. But the gametes separate independently of each other. So here's just an example of if we are actually crossing 2 different plants. So in this case, we're crossing, our P generation is made up of 2 true breeding plants, where if this purple flower self-pollinated, it would make identical offspring, which are all purple, and if this one self-pollinated, it would make all identical offspring, and that they're all white. Well, we're actually going to cross these 2, and you can do it with a nice little Punnett square, beautifully drawn, of course. And what you see in this is if we put our capital P's over here and our lowercase p's on the side, it doesn't matter if you have these flip-flopped either way. You would have capital lowercase, capital lowercase, capital lowercase, capital lowercase. So in this case, when we cross this P generation, 2 different true breeding plants, one purple, one white, we ended up with all 4 offspring being purple. Now, some of the terminology and names we give to these organisms when we're talking about their genotypes and their genetic makeup, if something is homozygous, it means it has 2 of the same alleles. That could be, let's say we're using the letter P, or the capital and lowercase letters don't look the exact same all the time. So if you're homozygous, you would either be capital, or lowercase, lowercase. Homozygous dominant being both capital, homozygous recessive being both lowercase. Heterozygous is where you have 2 different alleles. They're both the letter A, but we're going to have one capital, one lowercase. Your genotype is going to be your genetic makeup, and your phenotype is going to be your physical appearance. And I'll show you what that means here in just a second on the next slide. So we are going to do an example where we're going to cross a heterozygous organism with a homozygous dominant one. So with our Punnett square, I'm going to use P's to talk about flower color. Heterozygous organism has 2 different alleles, so it would be like a capital P and a lowercase p, with a homozygous dominant parent, so that would be a capital P and a capital P. Now you fill out your Punnett square like normal, so you have capital, capital, capital lowercase, capital, capital, undo that, capital lowercase. Now, we want to talk about our geno and phenotype. Our genotype is our different ways that we can have these letters arranged. So genotype is like, what do we see in our boxes? So we can either have capital P, or you can have a capital P, lowercase p. Genotype is a different combination of alleles you can get. Phenotype is what do these actually physically look like? Well, this flower right here and this flower right here are both purple, so 2 purple, and this one are both white, 2 white. So those are our geno and phenotype. Okay, I'm going to erase some stuff here. If we want to talk about something else we can do even further, is we can talk about our geno and phenotypic ratios. So let's do another Punnett square with the same example. We have a capital, lowercase, and a capital, capital. Fill out your Punnett square. So we said our genotypes in this case were capital P, and capital P, lowercase p. And our phenotypes were 2 purple, 2 white. Well, the ratio is a little different. Our genotypic ratio is what is the proportion that we have here. So our genotypic ratio here, we have 2 capital, to 2 capital lowercase, or we could simplify it and just say it's a 1-to-1 ratio. Our phenotypic ratio is going to be pretty similar. We have 2 purple plants for every 2 white plants, which also becomes a 1-to-1 ratio. So genotype, phenotype are the different combinations. Genotype's the different combinations of alleles. Phenotypes what they physically look like, and we can then put them into a mathematical ratio of our genotypic and phenotypic ratios. And we'll do another example here with seed color in pea plants in just a second. So something we can also do in genetics is we can actually do something called a test cross. If we have an unknown organism that we can physically see what it looks like. We know its phenotype, but we don't know the genotype. Let's say, for example, we have a flower that is purple. We know it's purple. We can see it with our eyes. But we don't know if it is homozygous dominant or if it is heterozygous with a capital lowercase. Well, in a test cross, you're going to breed this unknown with a homozygous recessive. And the reason is, if you breed something with homozygous recessive, you know what the outcome should be. So in the left example, if I breed a homozygous recessive with an unknown, and say the unknown was hypothetically homozygous dominant, I would capital P, lowercase p in every box, which would mean that if our unknown was homozygous dominant, you would have all 4 purple plants. If it was, hypothetically speaking, a heterozygous organism, we'd still fill out our Punnett square. And we would say, okay, well, the offspring of this one yielded something a little different because we had 3 purple and 1 white. So a test cross uses an unknown when you breed it with a homozygous recessive to tell you what the offspring are. And based on what the offspring are, you can actually figure out the genotype of the parent. And you can see that here from this example. It's exactly what we just did, except with now with colorful, beautiful pictures. If our unknown was crossed with a homozygous recessive, okay, what we would expect if it was a heterozygote was that we would have a 50-50 split of purple flowers and white flowers. If our unknown was homozygous dominant, all 4 are going to be purple because the dominant purple allele is going to overpower all of the white alleles and express itself every time. And when we're doing some of these crosses, there's a couple of ways to talk about it to determine what we're actually crossing. A monohybrid cross is where we cross 2 individuals that are heterozygous for one character, meaning they both have different alleles for their character. So it would be like if we crossed capital A lowercase a with another capital A lowercase a. They're both heterozygous for their trait. Now let's do an example in a Punnett square. Monohybrid, meaning one individual, so it's heterozygous for one trait with another. We're going to get A, and lowercase. Now, our genotypes in this case, just genotype, would be capital lowercase, or lowercase. Our phenotype would be, what do we physically see? Well, let's say A means if you're dominant, you're athletic. If you're lowercase, you got screwed in the genetic draft pretty much, and you are unathletic. Genetic lottery. So in this case, our phenotypes are we have 3 that are athletic, 1 that is unathletic. Okay, apologize for the penmanship. And let's look at our ratios. So when we look at ratios, let's do our geno ratio. When we do our ratios, we typically do most dominant to least dominant. So the most dominant square in this one we have is right here, followed by these ones, and followed lastly by these ones, or this one. So our genotypic ratio is going to be 1 to 2 to 1, because we have 1 very dominant here, 2 that are next dominant, and 1 that is lastly the least dominant. Okay, so 1 to 2 to 1 is going to be our ratio for genotypic. Phenotypic, what do they look like? Phenotypic ratio in this case, well, I have 3 that are purple, so those are all the same phenotype, and 1 that is white, which is not the same phenotype. So 3 purple are more dominant than white, a 3 to 1 ratio. So a dihybrid cross is a little more complicated. So in a dihybrid cross, we're crossing 2 organisms who are both heterozygous for 2 traits. So let's say we're going to use the example like Mendel does, and we'll see here in a second where we're talking about seed color and seed shape. So seed color, yellow is dominant to green. So this organism is heterozygous for yellow. And in terms of seed shape, typically round are dominant to wrinkled. So they're heterozygous for 2 traits, heterozygous for seed color and seed shape. So this organism is yellow, round seeds. And we're going to cross that with another organism who is yellow and rounded. And when you set up your Punnett square, it's actually going to be a 4 by 4. It's going to be 16 total squares. Okay. And the way you separate it to figure out what's on the top and what's on the side is a little different. So let's just look at what we have going on in this right here. Okay, let's say this is going to go to the top of our Punnett square. We have this, 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 and this, times this, 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 and this. Those are Punnett squares. So what you want to look at is what are the different combinations of letters I can have if I combine things. So let's say we're looking at Y's and R's. Well, I can technically have a capital Y and a capital R. I can also have a capital Y and a lowercase r. I could have a lowercase y and a capital R, or I could have a lowercase y and a lowercase r. So those different four combinations are how you set up your dihybrid cross. So in our dihybrid cross, we are crossing two organisms who are both hybrids for two traits. So capital lowercase with capital lowercase. Okay? And in these, what you'll typically see when you have a dihybrid cross is they call it a 9:3 to 3:1 phenotypic ratio. And let's look at what they physically look like. How many organisms do we have that are very dominant? Because phenotypic ratios, we go from most to least dominant. So how many organisms do we have that are dominant in their seed color being yellow and their seed shape being round? Well, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 that are yellow and round, because those are the two dominant traits. Now, how many do we have that are yellow and wrinkled? So yellow and wrinkled is 1, 2, 3. Then we're going to look at how many are green and round, because round is a dominant trait. 1, 2, 3. And lastly, how many are green and wrinkled, which is the most recessive? It's this one right here. Typically, you'll see we call a 9:3 to 3:1 phenotypic ratio in a dihybrid cross. A lot of what Mendel did laid the groundwork for genetics by telling us that usually traits are dominant or recessive, and the dominant one shows. But we have some complex inheritance patterns where we see that sometimes it's not necessarily dominant versus recessive, where capital P is always going to be purple, even if there's a lowercase p there. Sometimes we have different situations where there's not necessarily dominant or recessive. Sometimes there's more than two alleles. Sometimes there's more than two phenotypes. It can get more complicated. And these are some of the examples we'll see here today. Complete dominance we already talked about, where capital P means you're purple and lowercase p means you're white. So that's what we consider normal Punnett squares. Incomplete dominance we'll see is where neither of the traits is expressed, and codominance is where both are going to be expressed. So let's look at codominance first. Codominance, again, is where the situation where we're going to have both phenotypes expressed. Even though, in this case with cows, red is dominant and so is white, sometimes what you get when you breed them is this mix called a Rowan cow, which is red and white. When I was a kid, I always thought they were chocolate milk cows because Hershey's commercials brainwashed me into thinking that, but it is what it is. So let's breed these red and white cows. So we have a red one, which has two R's, and a white one, which has two W's. You end up in your squares, RW, 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 and they get this speckled red and white appearance. And it's called codominance because you see both the red and the white trait in the offspring. Both dominant traits are prevalent. Incomplete dominance is a little different. In incomplete dominance, neither of the original parent traits are going to be expressed when incomplete dominance occurs. So if we breed this red flower with this white flower, sometimes what you'll get is actually offspring that are pink in color, or they have the genes of both parents, they're heterozygous, looking something like this. But incomplete dominance means neither one truly shows. Instead of getting red or white, we actually get something in between, kind of like a blending. If you mixed red and white colors together, you would get pink. Incomplete dominance is that idea that neither one of the traits actually shows. It's a blending of the two altogether. Alright, so blood types are an example of something where we call multiple alleles. So in multiple alleles you have one trait, in this case it'll be one phenotypic trait would be blood type, is dictated by all the different types of alleles you can get. So what we see here is if you have type A blood, we typically see it as the allele is going to look like the letter I, and something followed by the I. So IA would be type A blood, as you can see here. Or you can also have I, A, I. The capital I's are dominant, the A's are dominant, the B's are dominant as well. The only recessive one is the lowercase I. So if you have type A blood, you're either IA, IA, or IAI. Okay? If you have type B blood, you're either IB-IB or IB-I. If you have type AB blood, you are IA-IB. And if you have type O blood, you are two lowercase i's. So what's interesting about this is both types A and B are dominant. And with your blood, if you notice these pictures here, okay, type A blood has these little triangles on them. Type B blood has these little circles. AB has both triangles and circles. And O doesn't have anything. So the way your blood works is your blood type has antigens and antibodies on them. That's what these little triangles and circles represent. And depending on what type of blood you have, it specifically determines what type of blood you can give or receive from people. So if you have type A blood, you can only receive blood from people who have either type A or type O. O is recessive. O is what we call the universal donor. If you have type B blood, you can receive blood from somebody else who has type B, or also from somebody who has type O. Again, O can donate to everybody. If you have type AB blood, you can receive blood from people who have type A, type B, and type O, because you have the antigen receptors and antibody receptors for everything, as we see here. So you can get from A, B, or O. And if you have type O blood, you can only receive blood from people with type O. So type O is the universal donor. Type AB is the universal recipient. You can kind of receive blood from anybody. What we say with these two, with your blood type, it's not super important to know about the antibodies, antigens right now. All you need to know is that we have one allele, or multiple alleles from the I gene here that determine one specific phenotype. Right, epistasis is another type of non-traditional Mendelian, or non-traditional genetic pattern where we have two genes located on the same chromosome, and one of them specifically affects the other. So let's have a perfectly drawn chromosome over here that has two loci, okay, and let's call them A and B, or B and E. So this locus here determines coat color in Labrador Retrievers. So if they have a capital B, it's going to be black, and the only way they can have a brown coat is if they don't have the capital B where they have two recessive. Okay, it could also be heterozygous and still be black, either way. So this locus here, this first one, determines the coat color. The second locus down here determines your deposition, or whether or not the color is actually deposited into the coat. So the second locus, you can either have a capital E, or heterozygous for your capital lowercase. And in both of those cases, they're going to deposit color into the lab's coat. If you are lowercase, you actually have no deposition. So there's not going to be any color deposited, regardless if it's black or brown or whatever. What you see in these is that the locus up here, locus number 1, we'll call it locus 1, is directly determined, or its color expression is directly determined by whether or not the locus down here has anything being deposited. And I'll show you here on the next slide. So with these labs here and their lab coat color, again, we said sometimes you have chromosomes that have two locuses or loci, or one will affect the other. And this top one we're going to say is for coat color, or you can either be BB, BB, or lowercase. And this bottom one is for deposition, where you're either capital lowercase, or lowercase. Now let's look at our labs we have over here. So if you have two capital letters, two capital B's, and two capital E's, you have the dominant homozygous for black fur coat, and you have dominant deposition, which says that yes, we are going to deposit the color into the coat, and it's going to be black. If you have a brown coat, you have two lowercase b's, which are recessive, so it's a brown coat, and two capital E's, which again says yes, we are going to deposit that color. Even if you're heterozygous, if you have a capital E at all, even if it's just one of them, you're going to deposit that coat into the color. Now, if you want a lab that is yellow coat, or yellow lab retriever, the only way you can have that is if your E for deposition is two lowercase, homozygous recessive, this one right here. So what that's saying is that this second locus is essentially like an off switch, which is inactivating the first one. So it doesn't matter whether or not we are this lab right here, okay, whether we are this lab right here, who has a capital lowercase which says he should be black in coat, since deposition is not occurring because we have two lowercase e's, it's basically like it's inactivating this locus over here and saying, I don't care what color genes we have, we're inactivating it and saying we're not going to deposit the coat. That's what epistasis is. Another example of different types of inheritance factors would be where you have polygenic inheritance. And in polygenic inheritance, you have two or more genes affect a single phenotype. So skin color. Skin color isn't just dark skin or light skin. Skin color is actually a play of a bunch of genes. So if you look at the scale over here on the right, individuals who have very, very dark complected skin are typically all dominant genes. And it's capital A, capital B, capital C, two of each. If you were the opposite and you had albinism or albino, you would be the opposite of that. You would be lowercase a, lowercase a, lowercase b, lowercase b, lowercase c, lowercase c. And this would be incredibly light complected. So polygenic means that you have multiple genes. So we have the gene for the A, the gene for the B, and the gene for the C in both of these dark or light complected individuals, that the interaction between these three genes affects the one phenotype of skin color. Polygenic multi-genes affecting it. And even though, like we just talked about with skin color, it's affected by multiple genes, there's also environmental factors that can play in. Norm of reaction and multifactorial effects are the idea that, yes, you may be dark complected if you have this skin color. Well, let's say you were something in the middle of the two extremes. So maybe you're lowercase a, capital A, lowercase b, capital C, let's say you are capital A, lowercase a, capital B, lowercase b, capital C, lowercase c, normally live in, I don't know, Georgia, Virginia, somewhere that's not too extreme temperatures. Let's say you move to Mexico because you want to get away. Okay, your skin color is probably naturally going to change as your body adjusts to the skin when you tan, when you're exposed to the extreme sunlight. So environment can also have an influence on certain phenotypes. Same can be said of red blood cell count. If people want to train for marathons, they want a higher red blood cell count, they can go train in the mountains. Lower air causes your body, or lower amount of oxygen in the higher altitudes causes your body to produce more red blood cells, which again, technically you have a genotype that determines how many red blood cells are in your body. But if you move or if you go train in these high altitudes, you'll produce more. So nature, nurture idea says that the environment can also affect your phenotype. This slide here is talking about the fact that typically recessive genetic disorders are but are brought about by heterozygous individuals who carry them. Since they're recessive, the only time you're going to have them, obviously, is when you don't have any dominant allele present. So heterozygotes are carriers because they're not affected, but if two heterozygous individuals end up having offspring, there's a chance that they will have that affected individual. And this is kind of one way that albino gene is brought about, is when two heterozygous parents or possibly homozygous recessive parents have children, they're going to give those recessive traits to their offspring. But typically, the heterozygous individuals are the ones who carry the traits. And you can have inherited disorders that are actually dominant. The case here would be achondroplasia or dwarfism. Dwarfism is actually caused by a mutation in the gene. So typically what you will see is individuals who have dwarfism are actually dominant, where it'll be capital capital or capital lowercase, but it's not very common because it arises by mutation. Most genetic disorders are recessive in nature, as we've seen, such as sickle cell, and the issue with malaria. But typically, the diseases are recessive. Dominantly inherited ones are usually caused by mutation. If two individuals who are dwarves or have achondroplasia have children, it's a 50-50 shot whether or not their children are going to be normal height or dwarves. This slide here just talks about some different ways where we can do genetic testing. Amniocentesis is one method where amniotic fluid is removed from the womb using a needle, or you can do chronic villus sampling where part of the placenta is actually removed to test the baby's DNA. They're both invasive. Techniques have improved, but neither one is really recommended unless you truly need to know what the genotype is of an individual who has not been born yet. And lastly, this slide, as well as the next one, I'm not really going to say anything over, but it's just a summary of all the different types of genetic inheritance patterns we've seen from Mendel, where it's either complete, incomplete, codominance, multiple alleles, pleiotropy, or it's epistasis, polygenic inheritance, or there's also environmental factors that play in. But this is chapter 14 in summary. Hope this helps.